Okay, get out these notes, everyone, if you would, please. In lab today, we do our lab, which is a fairly quick thing, and then you have your urinary system study guide, so you have plenty of time to get your questions answered. You're going to have to get that drink out of here. You'll have plenty of time to get your questions answered if you'll pull out and look over your questions on that. Everyone should already have a completed metabolism study guide, but if you have questions on it, do not hesitate to ask in that particular time. So Monday is your test on urinary system and metabolism and lecture. Everybody knows that, right? Okay. Do you have any comments to me before we get started? All right. We're on the urinary system, and we're trying to look at two huge hormone pathways that influence your urinary system. And if it's your urinary system, it's all about water conservation, saving water back in your body, not letting it dehydrate and leave your body in urine, so that you can maintain normal blood pressure. So these water balance for blood pressure. So this first theory is called the counter current multiplier mechanism. Let me get you oriented here. This is a very simplified nephron. So I've untwisted the proximal convoluted tubules. Here's your loop of Penley. I've untwisted the distal convoluted tubules. And what normally was wrapped around a nephron was paratubular capillaries. I have untwisted your paratubular capillaries and made them straight, for the reason that I want you to see what's going on between these two sets of tubes. Are you with me so far? And actually, from here to here, since I did not draw the glomerulus capillary bed, is really called renal tubules. But if I put the capillary bed in up here, now it quits being called renal tubules, and it is called a nephron, which it's labeled. So everybody knows that vocabulary? Now, and this is a collecting duct over here if you need to label that. You, or collecting tubule. You are called filtrate until you exit the collecting duct. Then and only then are you fated. It is your fate to become urine if you leave the collecting duct. So we're trying to see what's going on in here, and we are looking at salt concentrations, NaCl, because salt draws water. So in Anatomy 1, you talked about a concentration of a particular chemical when you were making formulas of solutions. You talked about a mole of something, and a mole was kind of like putting a cup of carbon in something, or a cup of sodium in something, except it wasn't really a cup, it was a certain number of particles, 6.02 times 10 to the 23rd particles. Well, this salt concentration here is being talked about in fractions of a mole. So we're talking about a lot less. These are milliosmoles of salt. But you can just think about it in terms of concentration. So as you look at this end of the nephron, there isn't a lot of salt in there. It's 300 milliosmoles. But look what it tells you salt is doing. There isn't much salt in the nephron, so salt is rushing in. And by the way, since on this side there isn't much salt, it's not drawing water, so water is exiting on this side where there isn't very much salt. But as salt continues to accumulate and accumulate and accumulate, you are going from, see the line? Once you're above the line, you're in the cortex region. Once you're below the line, you're in the region of the kidney that we call the medulla. So you're dipping down into the medulla, and then you're going back up into the cortex again. So by the time you get down in the deepest part of the medulla, look at the concentration of salt now. 1,200 milliosmoles of salt. A lot of salt down there in the bottom of your loop of Henle. Loop of Henle is labeled for you, and it tells you high salt. What would that want to do in terms of water? Attract water. And then you'd be putting out a lot of urine in your body. But lucky for us, this end of the nephron, where I have the dash, 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 is not permeable to water. So that is just a very fortunate thing for us that that end's not permeable, or we'd be rapidly dehydrating. Now, since you're at a really high salt concentration on this half of the nephron, look what salt's beginning to do. Exit back out to your tissue spaces. Let's look at this paratubular capillary. Now, do you see the arrows in the nephron? This shows you the direction the filtrate is moving from this end towards the collecting duct. Look in the paratubular capillary at the arrows. You are moving in this direction. So this is blood in the paratubular capillaries. Do you see that filtrate is going in this direction while blood is going in this direction? That is what a counter current is. It means two things going in opposite directions. It actually says that down here in first note at the bottom. A counter current is when the two things are going in opposite directions. Filtrate going an opposite way from the blood. What is being multiplied? The concentration of salt as you move towards the medulla. So that's why it's the multiplier mechanism. In both cases, the fluid, either filtrate in the nephron or blood in the capillary, is increasing its salt concentration to a maximum point as it flows towards the medulla. So let's see if that's true in the blood. We started out with 300 milliosmoles of salt in the blood, so salt is rushing in. By the time we get down here to the bottom of your paratubular capillary, you're at maximum salt concentration. And then look at this wonderful thing. The blood vessel is permeable to water. So since you have gobs of salt in the blood, this exiting end of the capillary is going to suck water out of the surrounding area, and you've just been dumping water out into the surrounding area from your nephron, and so we're restoring your blood volume right before we're back out here in the cortex becoming an interlobular vein. And veins aren't allowing transport in and out of any type. So we retrieved our water right before we turned from a capillary back into a vein, so nobody can steal it back from us, and therefore we maintain our blood pressure. So it's a really clever mechanism the way it works. So the bottom line is water is retained in the body, so your blood pressure is maintained. Now, this term right here, vasa recti, that's just what they call this section of the paratubular capillaries right down here by the loop of Henle. I'm never going to ask you about vasa recti. If you want to strike that out, that's fine. But it's just a special region of the paratubular capillaries when you're in the very straight sides of the paratubular, of the, loop of Henle. Now, I haven't even mentioned a hormone. So if we understand how this countercurrent multiplier mechanism works, the role of the hormone here is to get us even some extra water conserved in your body. And it operates right here at the collecting duct. In the collecting duct, there are pores. And those pores are never open unless your body is secreting antidiuretic hormone, ADH. Anti-diuretic diuretic hormone is ADH. It opens these pores, because otherwise, if it didn't, the filtrate would stay in the collecting duct and become future urine, and you'd lose it. So now it gets to run out, and that water stays in your body, and it'll eventually get to diffuse back into capillaries. So that's how you get even extra water conserved. So I want to look at this ADH pathway in detail, but in terms of your urinary system, this is the spot where it affects your urinary system. Okay. ADH stands for, on the next page, come on in, antidiuretic hormone. That was a neurohumor because it was made in a region of your brain, the floor of your brain called the hypothalamus. So this is a neurohumor that we heard about in lab on Monday. So the hypothalamus made it. And the hypothalamus is sitting there, remember, with the circle of Willis blood vessels pasted right against the hypothalamus like a postage stamp. So the hypothalamus is sitting there looking at your blood, and it's monitoring solutes in your blood, primarily sodium and chloride. So if your solutes get higher, that's implying you are dehydrated at the moment. You're lower in water, and so you have a higher osmotic pressure. You want to attract some water, in other words. So what does the hypothalamus do for you when it discovers this kind of situation? It fires, and it has been storing its ADH in synaptic knobs down in the posterior lobe of your pituitary gland, which was called the neurohypophysis or posterior lobe. And so when this fires, these synaptic knobs down here in the posterior lobe rupture, and it releases ADH. ADH is now released. So ADH diffuses into blood vessels nearby, and it does 4 different things. This is the thing we just found out it does in your urinary tract. It opens the pores of collecting tubules, and the body retains some extra water, and that'll eventually get absorbed into blood capillaries, and it keeps your blood pressure from falling. But 3 other things it does to up your water level is it makes you thirsty, it slows down your perspiration at your sudoriferous sweat glands, and it shuts the pores of your blood vessels so that you cannot be losing water from your blood vessels. And by the way, vasoconstricting the blood vessels helps your blood pressure too. So that's because it narrowed and crowded the blood more. So that's the second thing, is the vasoconstriction that helps you. What if you finally get out of this course in December and you're going to enjoy your holiday season and you drink too much liquor? What do you start doing? Urinating, 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 frequently urinating. The reason is a hypothalamus is inhibited by alcohol, so it's not firing. So if it's not firing, it's not rupturing the synaptic knobs in the posterior lobe of your pituitary gland, so you're not dripping ADH and saving water. Therefore, you've got those pores shut in your collecting ducts, and the urine goes on out of your body and takes with it all kinds of dissolved solutes. And that's probably why you feel bad the next day, because of all the minerals you've also lost at the same time. 2 other things that could stop you from making ADH is a tumor in the hypothalamus of your brain. This tumor is a mass, and it pushes on your nerve tissue in the hypothalamus, and so it doesn't fire. That's called diabetes insipidus, and the reason it is because you're not firing, you're not releasing ADH, you have frequent urination, and frequent urination is a characteristic of any kind of diabetes. So they name this tumor form diabetes insipidus. Has nothing to do with insulin, has nothing to do with glucose. It just is a form of diabetes because of this characteristic right here. What if your little kid is outside riding a bike and he snuck out there and didn't put on his helmet, and he clunked over as he was riding up the sidewalk and he hit his head on the steps leading up to your front door, wonked it pretty good, and you go out there and he's got a big goose egg on the side of his head. Actually, you hope to see a goose egg because that means maybe he's bleeding outside the bones instead of under the bones. But you're thinking, I don't know, I saw that was a head trauma, kabam. So you call your pediatrician and you talk to the nurse and you go, I don't know whether you want me to bring him in here so you can see him or not. And she says to you, is he doing anything different than normal? What she means is he frequently urinating, frequently urinating, frequently urinating? Because if he is, that means there's some pressure inside the skull up against the floor of the brain. So he must have a blood clot there, a hematoma pressing on the floor of the brain. Now he's not making his ADH because you've inhibited the brain, and so he's frequently urinating. That's what they mean. They don't mean is he suddenly flying around the house with wings. It's just, what is he doing that's health-wise not typical? Are we okay on that? All right. So that one's really not too scary. We mentioned these 4 things in lab, but now you see it relative to your urinary system. Okay. This is just a typed version of what I've just gone over with you, the countercurrent multiplier mechanism. Let me say one thing about this mechanism right here. If you're good about reading your textbook, there is another theory. It's called the 2-solute theory of why you conserve water back in your body. So some people don't believe it's just based on NaCl table salt. Some people think it's based on NaCl table salt and urea. Therefore, 2 different solutes are accumulating, and that is why you're retaining water. Well, you know, we know what urea does. Urea diffuses and hits a 50-50% relationship in your blood vessel and in your urinary system. So that's why a lot of people just go, well, that negates itself then, if there's an equal amount both ways. So, but if you read about it, so what? Maybe there is some urea, but there ends up being an equal amount both places. So more people tend to support this idea that it's more related to the salt concentrations than anything else. Okay, the last hormone pathway then of the urinary system is on the back of this page right here. Now, this is the aldosterone hormone pathway. So if you want to write that at the top of this sheet, then do so. Nevin, test Monday, urinary and metabolism. Y'all should have urinary study guides, the 2 of you, and you should have your metabolism study guide that we did together. And we have time in lab if you have questions on either one of those. All right, what I want to remind y'all of before we launch into this is, by the way, do not forget to study the black and white Xeroxes that I gave you of structures in a kidney and vessels of a kidney for this test, and structures of a nephron, etc. But when we got over to this view, I asked you to remember these cuboidal cells up here that are close to the glomerulus capillary bed. When you are close to something, you are juxtaposed to it, close, juxtaposed. So this is called the juxtaglomerular apparatus because it's a gland very close to your glomerulus. I'm fixing to mention it in this pathway. I just wanted to remind you, you had seen it briefly one time. It's called the juxtaglomerular cells or juxtaglomerular apparatus. It's a gland at the top of your glomerulus. So we will be talking about that. Folks, in addition to studying your paperwork from lab on pictures of kidney structure, study your urinalysis page. Not all the pages, just this page. Not the drips and what did we test for. I'm mainly interested in in these two left-hand columns. But if I gave you any vocabulary words like renal calculus, if I said know all of the URIA words, and if some of them are written over here in this column, then know any of the URA words like glycosuria or pyuria or hematuria or any of these. But I do not expect you to know all the abnormalities over here, just the URIA words, okay? But you should know normal urine and the test that you ran to test normal urine. So that's what those two left-hand columns are. So Lindsay, if you review this for your test, review these two left-hand columns and any vocabulary words that I stressed on it. Not so much the right-hand column other than URIA. And then know any picture pages that you have kidney structure on. And then this packet of notes. Now, are you ready? This is a little cheat sheet when we finish this story of the key chemicals, hormones in this pathway. But for now, let's walk through them and then we'll come back to that over on the left in just a moment. Obviously, we are watching water balance and blood pressure. And we're finding out something critical that tells the tale about how much water you have in your body is solute particles, which create osmotic pressure. So if you don't have enough salt, NaCl, then your osmotic pressure is going to drop and you need to save some more salt. Why? So you can save some more water. Why? So you can keep your blood pressure from bottoming out. So this is the theme in the urinary tract. So again, because of the circle of Willis blood vessels, the floor of your brain, known as the hypothalamus, is where both of these hormone pathways start. And of course, ADH wasn't really a hormone. If it was made by your hypothalamus, it too is a neurohumor. That one we just finished with, ADH. So that's our problem. We have a drop in sodium, which leads to a drop in chloride, which leads to a drop in water, and we have a drop in blood pressure. So now we're going to start firing the hypothalamus to correct these problems. This time, the hypothalamus is going to release a neurohumor made by the brain that is called ACTHRF, adrenocorticotropic hormone, ACTH, releasing factor. So this is a chemical that's going to make you release a hormone by this same name. It's just not going to have the word releasing factor at the end of it. Releasing factors only ever come from the hypothalamus. So if you're wanting to release a hormone from the anterior pituitary gland, then this had to release a releasing factor by the same name first. If you want the anterior pituitary gland to quit releasing a hormone, then you have to send out an inhibiting factor. It would be the same prefix and then it would end with IF, inhibiting factor. So hypothalamus controls the anterior lobe of your pituitary gland. So this is going to go in the blood and go to your anterior pituitary gland. And so it ends up here. So this is a cascade. Once one thing is released, it causes the very next thing to be released, and that causes the thing under it to be released. If anything in the sequence is not released, then this whole process stops immediately. So once you get to the anterior pituitary gland, which is also known as the adenohypophysis or pars discalis, it secretes a hormone named after the releasing factor. So it secretes ACTH, adrenocorticotropic hormone. But we're not immediately going to go to the adrenal cortex. It's going to take a few more steps, but ultimately that's where we're headed at the end of this story. That's why the anterior pituitary is your master gland, because it's going to control your adrenal cortex right here. Well, this travels in the blood and it goes over to your kidney. Each time it's going in the blood to the juxtaglomerular cells in the kidney. Well, there it says it right there. And it causes that gland to secrete renin. Juxtaglomerular apparatus secretes renin. It's called renin because you're in the renal organ, the kidney. Renin. Not renin. Renin has 3 N's. R-E-N-N-I-N. Renin is something in our stomach that makes our milk coagulate, especially in a newborn baby that's feeding on mother's milk. If it curdles and coagulates in the stomach, it'll stay in the stomach longer, so the stomach has a chance to digest the milk proteins and get that nutrient into the baby's bloodstream. So that's a different enzyme, but or hormone. This is renin, this hormone. So floating along in your blood are a couple of inactive chemicals that your liver made. Inactive but in blood made by your liver and dumped into the blood, and they've just been sitting in your blood for the day that you needed to run this aldosterone pathway. So look at the endings of these liver proteins. OGEN, that means they're inactive. So we've got to eat off an ending on each of these to get them turned on. So renin acts like an enzyme, with an IN like an enzyme. It eats off the end of angiotensinogen 1 to become active angiotensin 1. Angiotensin 1 turns around and eats off the little amino acid chain off of angiotensinogen 2, which is the N and the O are out of order, should be NO, to become active angiotensinogen 2. Now, this immediately is helping you with your blood pressure problem because angiotensinogen 2 causes vasoconstriction. So it is a class of hormones that we call a vasopressin. Anything that constricts a blood vessel is a vasopressin and is going to help increase your blood pressure. But the other thing that angiotensin 2 does is it also goes in the blood and finally makes it to your adrenal cortex, specifically to the outermost zona, which was called zona glomerulosa, and makes you release the steroid hormone from there that was in the group called mineralocorticoids. Its specific name was aldosterone. And this steroid hormone makes you save sodium, therefore you save chloride for free, so you use some energy to save the sodium. By an electrical gradient, you get the negative chloride for free, and therefore you get some water for free. So now you're helping to control water balance right there. You also save some negative bicarbonate ion, but because you saved all of this positive sodium, it repelled some positives out of the body, so you're getting rid of acid particles, hydrogen ions, and you're getting rid of positive potassium. because you saved this and got rid of this acid producer, you're also controlling your body's pH. So if you saved salt, that helped you with water balance. Because you saved water, you control your blood pressure and you don't bottom out. And you also help control pH. In this example, the anterior pituitary gland is producing the tropic hormone, which was called ACTH, because ultimately it's what's making this adrenal cortex in the end secrete its aldosterone. But you have to have all these chemicals in a sequence. The releasing factor from your hypothalamus, always. Then the next place you're going, if you have a releasing or an inhibiting factor, is to your pituitary gland. You release a hormone by the same name. Then that goes to your kidney, and so your juxtaglomerular cells secrete renin. Renin activates angiotensin 1. Angiotensin 1 activates angiotensin 2. Angiotensin 2 does two good things. It turns on the final hormone from your adrenal cortex so that you save the salt and save the water and regulate your blood pressure. But it also, angiotensin 2 is a vasoconstrictor by itself, so that action is going to elevate your blood pressure before you even get to the final step. So this is your little crib notes cheat sheet for a quick way of knowing that you've included all the key chemicals in this pathway. Are we okay on that? If you don't have any question on that, that is where your urinary discussion ends for your test material. So metabolism and urinary. If you look it over with your friends and you come up with questions, you can always come back in the second hour of the lab and get them clarified. Anything come up? You okay for the moment? Okay. Another announcement. Friday. I know I have gobs of office hours on Friday, but I've been put on 2 screening committees this semester. One of them I volunteered for, the other one they just put me on. But they are doing all this interviews for the potential candidates in the art department, so I'm not going to be in my office very much at all on Friday. So if you need to come see me, I don't have office hours tomorrow, but it happens that the 3 labs I teach in AP1 tomorrow are kind of short, like your lab today. So if you came in the second hour of any of one of those 3 labs and came into the 117 Anatomy 1 lab next door to your lab, you could ask a question if you get some confusions between now and then. Otherwise, write your questions down so when you see me on Monday, you can ask me. I have an office hour before your test at 11 o'clock. You could ask me then on Monday if you have questions. Just letting you know what's coming. Oh.
Jane, she actually had to go give her boyfriend.
Throw that. It's coming back. Pass that. Is there anybody else that needs one for somebody that's out? You know, there's some very wicked bugs out there right now that are hospitalizing students in your class or people's children with really severe fevers. And of course, we all know that there was a GI tract thing going around and the congestion respiratory thing. So try to get your rest so you can stay healthy. It is my intention to have this test on reproduction and hormones the Monday of Thanksgiving week, so that then you can go away for Thanksgiving and enjoy yourself, because I know what it's like when you all come back from that holiday. You've been hanging out with family, most highly traveled holiday, and it's like to get you revved back up for final lab exam and a final lecture exam, and then some of you have missed a test and you've got a makeup exam. I would prefer to get this one out of the way before you leave. So that's my goal. So you need to stay healthy, you need to not cut class, you need to get here, and when you're in class you need to be doing what we're on, not something else for another class or what you've missed because you cut class. So these are short little units we're on now. We get through them very quickly. They're not like digestive tract that takes a long, long time to go through, but you need to be here. Okay. And don't pull any boners. If you've missed a lecture test already, you can't miss another one. So don't pull something like that and think that's going to give you some special exception. The rules are clear. All right. Endocrinology and the reproductive system, and really these are almost impossible to separate because hormone pathways have so much to do with reproductive development. But we have already talked about most of the hormones, at least on a spreadsheet in the lab. And I am not testing you off of the spreadsheet in the lab. However, that's dangerous to say like that. In lab, you've been given another sheet that shows what the gland looks like and what its lobes are and what the name of its cells are. And that you do have to know for lab, because that's on either models in the room or it's on microscope slides. So that typical kind of stuff will be on the test. But as far as you telling me what happens, giantism is caused by an over-secretion, hyper-secretion of human growth hormone, that is lecture material. But the point is, when I'm talking about a gland and the name of the lobe and the name of the cell, that is the logical time for me to tell you what hormone it secretes. So we've already covered a lot of the endocrine stuff on that spreadsheet. So make yourself a note, know everything on the spreadsheet. This is what I'm talking about, you know, the spreadsheet. Know all of that for the lecture test. And so on that spreadsheet, I may have repeated the name of a gland or a lobe or a cell, but I also gave that to you on another stapled packet besides that spreadsheet so that you can keep one packet for lab. For lecture, you have to know the names of the glands and the lobes and the cells, though. So I've told you way back, whatever I gave you in lab that was structural, you still have to know for the lecture tests that are on that subject. So this is no different than normal. Endocrinology, we've had this definition in lab already, is the study of glands that don't have ducts to their target site, and so their hormones diffuse into the bloodstream, and then the blood carries them out to their target tissue, and they diffuse out of the blood to their target organ or target cell. The word hormone means to excite, and so the reason you are secreting hormones to go to a specific organ or a general tissue of the body is to get that organ or tissue back near balance, internal balance, which is known as homeostasis. We probably never perfectly get there, but your body's always trying to get back to homeostatic balance. And when we're very much generalizing, we call all kinds of things hormones in an umbrella sense of the word, like all of these things listed right here. But truthfully, these things listed right here are made by nerve cells somewhere in your body, either in your brain, like the hypothalamus, or and these are in the hypothalamus, or in the nerve tissue in the center of your adrenal gland, known as the adrenal medulla, that acts like a sympathetic nervous system reaction. So all of these things in Roman numeral 2 here are neurohumors, including the regulatory factors that are either releasing factors RF or inhibiting factors IF from your hypothalamus. So if you want to clarify this, the ones that are underlined are from your hypothalamus. The one that is circled is from your adrenal medulla. So they aren't really hormones, but so if I asked you what are they, you wouldn't write hormone, you'd write neurohumor. But we always study them when we're studying hormones. The underscored ones come from your hypothalamus.
What was the circle one?
The circled one, it tells you, came out of your adrenal medulla, and that's when the adrenal medulla is thought to be sympathetic nervous tissue acting in the fight, fright, flight scenario. So it'll speed up your heart rate, speed up your blood pressure, speed up your respiratory rate, increase your blood flow to your skeletal muscle, shut down your skin, cold and clammy skin. Are we okay? A tropic hormone is a vocabulary word. It comes from one real gland and makes another real gland secrete. So all of the hormones from your anterior pituitary gland are considered tropic hormones. So some examples that we'll be talking about more as we get to reproduction is your anterior pituitary made follicle stimulating hormone, luteinizing hormone, and interstitial cell stimulating hormone. These are ones that have an effect on the ovaries of women or the testes of men. For example, it makes the ovary make estrogens and progesterone, and it makes the testis make testosterone. So these are your gonadotropins coming from your anterior pituitary to go to these red gonads.
Do we need to know the full names?
Eventually, yeah, you're eventually going to have to know the full names. You have them already on your spreadsheet. We've been over them. But this is just a quick reminder of some examples of some tropic hormones that we've touched on. But FSH is follicle stimulating hormone. And then in the woman, and that's in male or female, luteinizing hormone is only in the female. That's luteinizing hormone. And in the male, a similar chemical is renamed ICSH for his cells that make testosterone. Those are interstitial cell stimulating hormone. And so when these three gonadotropic hormones go to your ovary, it makes the ovary secrete E, which throughout these notes is going to be your estrogen, and P, which is progesterone. Estrogen and progesterone are female sex hormones. And T from the testis is testosterones. Those are not gonadotropins. Those are what are released because your ovary and testis were stimulated by the anterior pituitary glands gonadotropins over here on the left. Ovaries and testes are your gonads. What's a gonad? The organ that makes sperms and eggs. That's what makes you a gonad. Did that answer all of that? Okay. So we classify hormones by several different ways. We classify them by hormone types, like do you circulate in the bloodstream or do you not? And most of the ones we concentrate on are in the I do circulate around the body in your bloodstream. So they travel in the blood, they're going to a specific target cell, target tissue, target organ, and if they're going to a lot of a particular type of tissue, like human growth hormone makes cells in general grow, then it could be having a somewhat general response in the cells throughout your body. But a lot of times, as in the case of these gonadotropins, they're going to a very specific target organ, ovary or testis. So how specific it is can vary from hormone to hormone that's traveling in the blood. But those are the circulating ones. Let's say I'm a body cell. So, there are other hormones that a body cell makes just to control the boring daily activities of that body cell, like how you get food in, how you get waste out, how you control certain metabolic things, like are you getting too acidic or too basic. And those are non-circulating hormones because they don't go from that cell into the bloodstream. They stay right there. Those are fatty acids. They're not even full-blown lipids, and they're known as prostaglandins. But we don't do much with them, but they're really, really important because most disorders in a body ultimately come down to some cell isn't doing what it should do. It's either doing something too much, or it's not doing it at all, or not enough. And so if these are chemicals that regulate the housekeeping daily activities of a cell, then probably prostaglandins might be involved when a cell is dysfunctioning, becoming diseased, aging. So they think we'll be looking at these research-wise more and more and more as years go on. If you only control the very cell you were made in as a prostaglandin, you're called an autocrine prostaglandin. If you go and maybe diffuse into the cell right next door or a couple of cells right around you, but you didn't go in the blood, you're still a prostaglandin, then you're called a paracrine. So they divide the prostaglandins into two groups, whether they just stay in the cell where they were made, autocrines, or whether they go to nearby neighboring cells. But they don't go very far. This part right here, membrane associated, we'll come to in a little while, but here's the deal. Some hormones control their target organ by entering the cells of the target organ and even going all the way into the nucleus to the DNA genes. Other hormones glue on the outside of the cell at what is called a cell receptor site, and just by plugging into a receptor site, it tells the cell what it wants you to start doing or quit doing. This one gets stuck on the outside. It's membrane associated. It doesn't even get to go enter that neighboring cell if it's a paracrine. If it's going over to a neighboring cell, it just goes to its membrane and plugs into a receptor site. We'll talk more about some of that later. But it alters the chemical reactions, which are known as metabolic reactions of the cell, telling it to start something or stop something, speed it up, slow it down. What makes your body release hormones? Sometimes it's a change in body chemistry. In the case of male sex hormones, from puberty to the day they die, they're supposed to have X amount of their male sex hormones, their testosterone. So if chemically, if they start declining in the amount, then that's what triggers their testes to secrete some more. End of story. That's how they work. So sometimes in your body, it's just a change in chemistry. Other times it's pressure, mechanical stimulation, like the birth hormone, oxytocin, at the uterus or breast of a woman. That was a pressure story. Other times, some foreign agent has entered your body and you're making the hormones in response to that. Are we okay on this so far? All right, so we sort of classified them whether they go in the blood or they don't go in the blood. Sometimes we classify them by their chemical structure. And if I haven't given you a hormone in here, I'm not expecting you to go look it up. I'm just showing you how hormones behave and giving you a few big examples. Who cares what their chemical structure is? The reason you care is because some hormones get to come in a cell and some get excluded and blocked out of the cell and are just membrane bound. What would you rather be in order to gain access into the cell? Well, it all comes back to Human Anatomy and Physiology 1, and what was the main structure of human cell membranes? A cell membrane is a double layer of phospholipids, lipids. So like dissolves like. If you want to get in a cell, you better hope you are probably a lipid, fat soluble. The majority of those get like a cholesterol or something like that, steroid. Those usually get in. Usually, if your amino acids or something that's like an amino acid but it's lost in oxygen, it's called an amine, or a little chain of amino acids, polypeptides or protein, usually these don't get to come in. They usually don't. There are exceptions. We just told you one exception. Here's something fat soluble, and yet it did head it at the end of it. Didn't get to come in. It got excluded to the outside of the cell. So prostaglandins are weird in that sense. They weren't permeable through a membrane. But most of these don't get to come in. There are always exceptions, but anyway. Where do you make a protein? Where was your protein factory from Anatomy 1? Which endoplasmic reticulums? Rough, because you had to have ribosomes. So your protein factory is your ribosome. That's why these protein-like ones are made on rough ER, because rough ER has ribosomes studding the edges of the endoplasmic reticulum. And then once the protein hormone is made, or protein-like hormone is made, the endoplasmic reticulum tubules are not static. They can remake connections to other organelles. So they make a connection to your Golgi apparatus, Golgi body, and dump the protein-like hormones into the Golgi, and then the Golgi body concentrates them and pushes them out to the end and wraps them in little secretory membrane-surrounded vesicles and spits them out of the cell. So if you're a secretory gland, you probably have a lot of Golgi bodies per cell. But that's how they're transported, is through the ER to the Golgi and then spit out of the cell. So here are some examples of ones that are protein-like in some way. And notice that every single one from your anterior pituitary gland is protein and going to probably be kept on the outside of the cell most of the time once it's made and secreted from its gland cell. So the other category are the lipid soluble group, and those are the steroids. And you're called a steroid if you're made from cholesterol. They think that these are made in the smooth endoplasmic reticulum or the mitochondria. You don't tend to make extra of these. You make them as you need them, and so they don't tend to be stored. And I may have mentioned this in the class. That way, if you cheat and you take extra steroid like testosterone, they can tell it because it's not normally stored. And if you're taking extra doses like veterinary testosterone so that you have more muscle and you can win a contest, then you're going to have levels of that showing up in your bloodstream that are more than the average individual, and it's very detectable. These steroids are your sex hormones, like your estrogens and progesterones, your testosterones, and the 3 steroid groups that come from the zonas in your adrenal cortex. That's where the steroid ones come from. And those are usually the ones that enter the cell and go into the nucleus and turn genes on or turn off genes. We'll come back to that. So here are some examples of some that we have studied that are from the adrenal cortex. Eicosanoids is the fatty acid ones. In other words, these are also lipid soluble, but they're an exception to the rule. Although they're made of fatty acid, they don't get to enter the cell of a neighboring cell. They operate from outside the cell. We'll just say operate on cell membrane. Okay, so we've mentioned where some of these are made, ribosomes versus mitochondria or smooth endoplasmic reticulum. We tried to point out why their chemistry is important. You get kept on the outside of the cell membrane, or you get to come inside the cell. So now we've made them and we've secreted them. Where are they now? They're in the blood. So we want to understand how they're traveling along in the blood. This is what this part is about. So this is the mechanism of transport in your bloodstream. And the solute, the protein solute that is most abundant in your blood, the protein, is albumins. And they give blood its viscosity, its resistance to flow. But why the albumins are important now is they are carriers for endocrine hormones in your bloodstream. Not every single endocrine hormone will bond to an albumin. Half of one will. So if I just secreted testosterone, then half of this testosterone that's in my bloodstream will be hooked to an albumin, and half of it will be free floating in my blood plasma. So that's what this means. Half of the hormone floats around, the other half is bonded, bonds half of the hormone to the albumin. Who cares? Because under normal circumstances, albumins are huge proteins. They don't get to leave your bloodstream. So the only hormone that would get to go out to your target tissue is which one? You got bonded to albumin hormone, and you got hormone floating freely in the plasma. The one floating free in the plasma, because it's not hooked to a huge protein that keeps it from being able to go through the pores of a capillary. So this one floats free. The free hormone is what diffuses out to the target site. And then that means you don't have any free floating hormone right now. So then all of this corrects itself. Now you've got half of the original amount left in there. So it will become half of that bonded to the albumin and half becomes free floating. You know the beauty of this? It means every time you go around your body, let's say I just started secreting a hormone to change something. If I just started secreting the hormone, then I don't have a ton of it in my bloodstream. It's just started entering my blood. So a little bit is bound and a little bit is free. And so it diffuses out to its target tissue, and it's starting to tell that target organ, we're going to begin to do this. And then the next time my blood flows around my body, because all your body blood flows around your body about every 1 minute, so the next time it flows around, I'm probably accumulating more of that same hormone. And so I'll get a bigger flood of it going out to the target organ, which will speed that target organ on its way to doing what it's supposed to do even more. So what you're doing is you're very slowly ramping up the activity level of the target organ. And then when you want to stop it, you don't just go like a light switch flipped off. You tamp it down slowly by sending less and less out there. So you're not shocking an organ or a tissue. It's a matter of, are we stimulating or are we inhibiting, not on and off. You get what I'm saying? Is there anybody confused about this? Okay. Well, now we're going to look at the ones that don't get to come into the cell. These are circulating hormones. They're in your bloodstream. They've made it out to their target cell. And so when they're there, these are hormones that act at specific fixed receptor site on the outside of the cell membrane. And those receptor sites from Anatomy 1 were made of peripheral proteins and carbohydrates. That's the structure you studied in Anatomy 1 that made receptor sites on the surface of your cell. Proteins and carbohydrates. But anyway, what does fixed mean? It means if I make this be a cell, if I make this be a cell right here, then if on the surface of this cell there are different shapes, then maybe right here there's a receptor site that invaginates like that, like a little rectangle right there. That's one receptor site. So what I'm doing right down here on your handout sheet is I'm magnifying that little section of a cell membrane. And so here is your cell membrane. There is the rectangular receptor site I'm referring to. It is called fixed because that rectangular plug-in site doesn't move from spot to spot on this cell membrane. Wherever it is, it's always right there. That's what they mean by a fixed receptor site. So in this example, this right here is your fixed receptor site that we're referring to. So you have different hormones floating by. You could have a hormone shaped like this. You could have a hormone shaped like this. You could have a hormone shaped like this. Well, which one of these hormones is going to fit in that specifically shaped receptor site right there? First, middle, or bottom? First. They have to be complementary. So just like your key in your front door, that will fit into that. So they're showing this move over and plugging into its receptor site. That's what's going on there. And this mechanism of plugging in on the surface receptor site is used by most of the amino acid, amine, protein, and prostaglandin type hormones. Those are the ones that behave this way. If you don't like the name specific fixed receptor site, another name for that you may have learned in Anatomy 1 is called a G protein linked receptor site. That's another name. So it says AKA, also known as a G protein linked receptor site. But you see their shapes have to be complementary or it'll never plug in. Now, in this story, this is now the inside of the cell over here. And out here, you're on the outside of the cell. And it says that right here. The protein is known as the first messenger. So in notes down here, you have that going on. In the inside of the cell membrane, this edge right here, which corresponds to in here, the inner edge, there is an enzyme there. The name of the enzyme is adenylate cyclase. If you're using a much older textbook, it might just call it adenyl cyclase. But adenylate cyclase has to get turned on. The way it gets turned on is by the first messenger, the hormone, plugging in to its specific fixed receptor site. That then turns on the enzyme that's been sitting here in an inactive form. So now we have that turned on. What does adenylate cyclase do? It breaks down ATP, but it doesn't just break it down to ADP and phosphate. It breaks it down and breaks 2 phosphate groups off of it, so it becomes cyclic AMP, adenosine monophosphate. It broke 2 phosphate groups off of it. Who cares? Because this cyclic AMP acts as the second messenger. That acts as your second messenger, and it is able to travel in the cell to cause whatever you wanted. See, it says second messenger. It causes whatever you wanted to happen in the cell due to that protein being there to happen. So the example of the hormone that we're doing with this is glucagon. There was glucagon and insulin. What did glucagon do? It did what? Yes, and built it up in your blood. So I'm just going to simplify what you said and say this raises blood glucose. There's a fruit fly in here. Trying to take it back. So we know that that's what that hormone does. Well, the way cyclic AMP carries out the role of the glucagon, so I'm telling you that this hormone right here that's plugging in is glucagon. That's your first messenger, but it didn't come in the cell. So the second messenger carries out the role of glucagon. So it comes over here in the cytoplasm, and it, at the ribosome, it phosphorylates something. In other words, it turns on some reactions by turning on enzymes. Well, let's say you're a liver cell, and glucagon has come to a liver cell. There's glycogen starch stored in a liver cell. Is it going to make you make more glycogen starch out of glucose? Or is it going to make you take glycogen starch and break it down to glucose if what you're wanting to do is raise blood glucose levels? It's going to want to break it down. It's going to want to make lots and lots more glucose so you can dump it in your bloodstream and do the role of the hormone. So literally, cyclic AMP comes over here and it activates this side. This is the go side. It activates the enzymes that will break down the glycogen and turn it into sugar, and it inhibits, it says inactivates, but this is it inhibits this side, so you're not stealing the glucose out of your blood and converting it back into the stored glycogen starch. That's why it's acting as the second messenger. And most of the time, the way second messengers work to turn something on is they stick a phosphate group on it, and that's what turns it on. There are, however, a couple of exceptions to that, and I'm going to talk you through a couple of the exceptions. So, if I were the second messenger, what are some of the things I could have triggered? In this case, I did that one. That's the one we just talked you through. But if I were some other hormone, I could cause muscle contraction somewhere. That might be my role. Or I might be a cyclic AMP that activates genes so that you make DNA into RNA to build a protein that then makes you do the role of the hormone in the body. Or I could cause secretion by glands. Or I could change the permeability of a cell membrane so that something that was getting in now does not, or something that wasn't getting in now does. So it all depends on which receptor site on the surface of the cell I plug it into, whether I know to do this or this or this or this or this. You don't just pick one. It's all determined by what the surface receptor was. Let's look at the opposite of glucagon. Glucagon was the first messenger, turned on adenylate cyclase on the cell membrane, which broke down ATP to make your second messenger, cyclic AMP, and the cyclic AMP made you break down glycogen starch and get a lot of glucose in the blood. This is insulin. This is insulin that's going to be the first messenger. Insulin doesn't have a second messenger. The hormone acts as the second messenger and adds a phosphate group. And what this does is there were some pore channels, big glucose needed a pore channel to go to get through this cell membrane and enter this body cell. And that's why insulin is known to lower blood glucose. But in order to, we'll just do this, to open the glucose pore channels, the insulin had to phosphorylate a compound on the surface that was keeping those channels blocked. So it was acting as the first as well as the second messenger. The only other thing that'll open those pore channels is exercise. That's why if you are a borderline diabetic, your doctor says you need to exercise. Yeah, they're always hoping we'll lose weight, but that is not why he's telling you to exercise. If you exercise, it'll open those pore channels without insulin even being there, and your body cells will get some sugar inside, and it lowers your blood glucose. So if diabetics understood they're helping themselves lower their blood glucose, they would take the walk around the blocks and the things that their physician is asking them to do. But that's an exceptional one that doesn't have a second messenger. Finally, we've already talked about oxytocin, but in oxytocin, this is an example of one that causes muscle contraction. Oxytocin is the first messenger. Its second messenger is an exception. Its second messenger is calcium. And the more calcium that you have released, the more inhibitors you can pull off of the actin, and the more sliding of the actin and myosin to cause uterine contractions you can have. So here's the second messenger here that's weird. But most of the rest of them act with the adenylate cyclase and the cyclic AMP. We'll stop there. Next time when we come back, we'll talk about the hormones that get to come inside the cell and operate at genes. And we'll be moving on fairly quickly to reproductive tract.